Okay, so I'm a returnee. This is my second talk on this topic, um, and the topic has created quite a lot of interest because really, as we go back to work and we start to move closer and closer together again, uh, the, we don't have that many options for limiting airborne viral transmission from person to person. We've got masks, we've got social distancing. Far-UVC light was designed as a third possibility in our battle. So, yeah, we've been getting an awful lot of questions about what exactly is Far-UVC, how might it be used. So I'm designing this talk as a kind of FAQ as to what we see as the potential in the future. And this is my favorite question. Came this last weekend. Will my UV sensitive tattoo protect me from COVID-19? Well, I didn't even know there were UV sensitive tattoos. But yeah, it moved me to think about a Far-UVC FAQ. And I think the question we are asked most often is, well, we know about conventional germicidal UV light, but is Far-UVC light in some way different? What is the difference? So here is the spectrum of UV light right next to the visible light spectrum. And UVC you see on the left between 100 and 280 nanometers. Conventional germicidal UV light, which we've known about for a century, basically is around 250 nanometers. And Far-UVC light is just a different wavelength. So it's around 220 nanometers. So you might think, well, that's not so much difference. So how come it's a significant difference? Well, first of all, both of these types of UV light kill bacteria and viruses very efficiently, and I'll come back a little bit to that, including coronaviruses. The difference is in safety. So Far-UVC light, 222 nanometers, has a very, very much shorter range in biological material than conventional germicidal UV light. And what you see on the right-hand side here is an absorption spectrum, UV absorption spectrum, basically in protein. And what you see is that the 222 nanometer light is very efficiently absorbed in protein, so essentially in all biological material. And that's very different from 254 conventional germicidal UV light. So what that means, what the significance of that is in terms of safety, is that Far-UVC light, the 222 nanometer light, really can't penetrate the dead cells at the surface of our skin, the stratum corneum. And so it basically can't reach any of the living cells in our skin, in the epidermis or in the dermis. And the same applies to the eye. And the eye, instead of the stratum corneum, we have the ocular tear layer, which is right at the very surface of our eyes, and so prevents the 222 nanometer light from reaching the cornea or anything deeper than that. And that, again, that's as opposed to conventional germicidal UV light, the 254 nanometer light, which certainly can penetrate the stratum corneum, so can reach the epidermis and the living cells in our skin, and likewise can penetrate the tear layer and reach the cornea. So what that means is conventional germicidal UV light, the 254 nanometer light, has its role. It's used, as many of you probably know, in many hospitals to sterilize operating rooms and surgical equipment. And very much more recently, the conventional 254 nanometer light has been used in the MTA, in subways, or is being used in subways and in buses to sterilize the subway trains and buses. But so here's a picture of that, and what you see is there are no people here, and that's because the 254 nanometer light presents health hazards to the skin and to the eye. So the MTA, at least temporarily, is closed down between 1 and 5 in the morning. So they're taking that opportunity to sterilize the trains and buses overnight, and that's what you see here. Of course, come 5 o'clock in the morning, when hopefully you've got a nice decontaminated subway train, people start coming in, and all your losses or your gains are gradually lost over the course of the day. So the notion then is that Far-UVC light is, quote, safe for human exposure. So it can be used actually while people are present. And so we don't have to, we worry less then about recontamination during the day. So of course the big question, which is the main question we always get asked, is well, how do we know if Far-UVC light is actually safe? And I would typically give three answers to that question. One, it's the biophysics, and in general, I would say biophysics is a more reliable indicator than biology. It's more repeatable. The biophysics says that the UV light simply can't reach the living cells in human skin or the eye. Obviously, that's not enough, and there have been many studies over quite a few years now, both here at Columbia and in other places, with human skin models, with human skin itself, with mouse skin models, and with mice, to look at safety studies. And really, they've been all very encouraging, that they back up the biophysics and show whenever you do a head-to-head comparison of conventional 254 nanometer light and 222 nanometer light, you see effects with 254, but you don't with 222. And the last point I'd like to make there is that actually there is an existing national and international safety regulatory framework, which basically says at this moment how much UV light is allowed for human exposure during the course of an hour or a day or a month. And this graph here actually shows what the daily limit actually is. And so I think all the manufacturers who are starting to deploy these lamps are going to stay within the regulatory framework, although the regulators have talked a lot in the past month or two about relaxing those regulations for 222 nanometer light. But I think it's fine to stay within the current limits. So the next biggest question has been, well, what exactly are you thinking that Far-UVC light will do? Is it aimed at airborne mediated transmission, or surface mediated viral transmission, or both? And I think all along we have thought of it in terms of limiting airborne transmission. And certainly as the months of the COVID-19 crisis have gone by, there's been a different perspective, I think, on what exactly is the main mode of transmission. I think initially it was thought to be surface mediated, but more and more I think it's now being thought to be primarily airborne mediated. So what you see in this little table here is a very rough estimate of if somebody coughs and sneezes and a whole variety of particles and aerosols comes into the air, what are the sizes and what are the times associated with that? So the bigger particles are affected by gravity, and they fall to the ground very quickly in a few seconds or perhaps a minute or two. The smaller particles, on the other hand, they stay, they're not affected by gravity, and they're going to stay in the air pretty well indefinitely. And those are the ones we worry about most in a scenario where people are together in a room and potentially coughing and sneezing. So in the end, you're going to get an ambient level of these small particles, some obviously potentially containing virus, that are in the air and are just going to stay in the air. So the notion behind Far-UVC lighting really is to try and reduce that ambient level of in our case, COVID-19 in the air by continuously exposing the air to Far-UVC light and trying to take a bite out of those indefinitely suspended particles that are in the air. So, an obvious question, well, do you know that Far-UVC light will kill coronaviruses? Well, I mean, I think we would have known that really without doing any experiments at all, because UV light is well known to kill all viruses with considerable efficiency. But published actually, I believe it was yesterday, this first paper on Far-UVC light efficiently and safely inactivates airborne human coronaviruses. So in this paper, we've basically used a couple of the seasonal coronaviruses simply because it was easier for us to do. We can do it in a BSL-2 facility, and we were able to do that pretty quickly. And basically, the experimental apparatus is here, and you can see we flow. So virus starts on the left-hand side. It's aerosolized. It flows in front of this window, and there's a Far-UVC lamp facing this way, just for the photograph. And then on the right-hand side, we collect the viruses and we assay them for inactivation. And the results from those studies suggest that at the very low levels of Far-UVC, continuous Far-UVC exposure, and those are certainly within the current regulatory limits, you'd get about 90% inactivation in 8 minutes, 95% in 11 minutes, 99% in 16 minutes, and 4 logs, 99.9%, 3 logs, sorry, in 25 minutes. In other words, what you're trying to do by having this Far-UVC light in public indoor locations is just to lower the level of the potential level of ambient COVID virus in the air. You're not in a few seconds trying to kill the viruses. You're steadily trying to reduce the ambient level in the air. So the next question would be, well, those are the seasonal coronaviruses. What about the SARS-CoV-2 virus? Is that the same? And again, without doing any experiments, you would have said yes, because really the UV sensitivity to different viruses is not that different from one virus to another. It varies some, of course. So those experiments can't be done in a standard BSL-2 facility, and Ian Lipkin's team kindly took that on in his BSL-3 lab using real-time PCR as opposed to the standard dilution assay that we've been using. But the results appear to be similar, and current results suggest about 4 logs of inactivation after a pretty low dose of 5 millijoules per centimeter squared. So I think it's clear that it will work for all viruses. And I mean, that's a more generic point, because a question often asked is, well, is this specific to the current crisis, or is it going to work for the things that we worried about before, influenza, measles, TB? And what about the next pandemic virus, which we hope won't come, but probably will come? And again, I think the answer is yes. This is a generic approach to killing viruses and airborne viruses and bacteria. And the UV doesn't really care about all the technical details of the proteins and the receptors and the like. All it does is damage the DNA or the RNA in the microbe. And every bacteria and virus has DNA and RNA, of course. So the study that I'm showing here is one we published last year, which was on H1N1 influenza virus. And again, showing that Far-UVC light efficiently kills airborne H1N1 virus. And then the big question, well, how do you see it being used, and where do you see it being used? And we've had just an incredible amount of suggestions and positive proposals, and this is where we should be using Far-UVC light. And you know, it's basically anywhere where where people, indoor locations, where people are coming together, and as there will be more and more in the future months, and hospitals, obviously, and dental hospitals, but transport, any transport facilities, restaurants, offices, schools, theaters, shops, gymnasiums. I think all these types of facilities are interested in another methodology for trying to reduce airborne viral transmission beyond social distancing and beyond masks. And coming home, well, what about NYP and CYMC? Obviously, we've had a lot of interest in installing these devices locally, and that's going to happen. So coming very soon, the first demonstration project is going to be at the NYP Gamma Knife facility, which is NYP Radiation Oncology. Chosen really because it's a moderately small facility, and we can control things well, and we plan to do obviously a lot of testing to see if we are reducing the microbial content in the air. And we think that will happen really very shortly, in the next few weeks. And the College of Dental Medicine is obviously very interested in this, because aerosols are a major issue for in the dentistry world, as you might imagine. So in the planning stage is another demonstration project in the College of Dental Medicine here. So let me finish by.
You have 2 minutes. Yeah.
Yeah, just mentioning where we think the future developments lie. Well, one is with the lamps. So the lamps are gas-based lamps, and they work pretty well. They're moderately expensive, a few $100, and the production will be in the hundreds of thousands and millions towards the end of the year. But the future, we all think, is in LEDs. LEDs are easy to make, cheaper, longer lifetime. There are issues at 222 with at these sorts of wavelengths. The efficiency of the LEDs drops like a stone, as you can see in this graph here. So there needs to be some development from the LED manufacturers, and that's certainly happening. Installing and testing Far-UVC lights, demonstration projects, and a lot of pressure to actually start installing them in real-world situations, restaurants, for example. And then on the research side, I mean, we need good airborne transmission animal models to better understand, well, what are all the environmental factors that are going to affect this. And we had just started actually these studies before COVID started with influenza and a guinea pig model. For the COVID-19, well, ferrets perhaps, Syrian hamsters, but BSL-3 animal facilities for those is going to be difficult, I believe. So perhaps the future is going to be with the humanized ACE2 mice that we heard about in the last talk. And the general idea you see on the right here is you have a cage with infected animals and airflow to a cage with non-infected animals, and with and without the UV lights, you see whether or not the recipient animals get infected. So I think my time is up. I will stop there, and I'm happy to answer any questions.
Terrific. Please, again, raise your hands if you have any question. I see three. Neville?
Yes, hello. I was just wondering, maybe about the use of eyewear under controlled settings that might reduce the exposure in dental clinics, et cetera, until all safety analyses are complete.
Yeah, and there's absolutely no reason not to do that, and it makes sense. Again, I mean, we have a long-term study right now with 100 mice who have been exposed to, getting on for a year, to very much higher exposures than we're potentially going to use for beings. And after, I think it's 42 weeks and getting monthly eye tests, none of them have any evidence of any problems there. But yeah, of course, there's no reason at all not to wear eye protection, and just regular glasses would work as well.
Great. David Cohen?
Yes, thank you for this really interesting talk. Two questions. One is, how far from the light emission source is the Far-UVC effective? How close does it need to be? If you're thinking of a train station or something like that, where it might be a very large area. And secondly, would you foresee any additional safety concerns with children? Thank you.
Let's see. Well, your first question, I mean, the lighting, how you design the lighting is very similar to the way you would design a lighting, visible lighting. So an architect having a room that they're building and they have to figure out how to light it. Well, there are professionals who figure out how to produce a given level of desired level of lighting in the room. And it would be exactly the same thing for Far-UVC. So basically, one anticipates that these lights would be coincident with the visible lights, and they would be placed in the same locations. Far-UVC light is not absorbed particularly in air, so there aren't too many issues there. It's just a matter of deciding, well, this is the level that I would like, shall we say it, at head level for the occupants of the room, and designing where to put the lights on that basis. And in terms of children, the issues are not different for children and adults, because the stratum corneum for children is not systematically any different from adults, and the same with the tear layer. So if it's, I don't think there are age-dependent issues of safety.
Great. I think we have Rodney Rothstein.
Hey, David, thank you. That was great. I wanted to ask a question about wound healing. During healing in the skin, are there times when cells would be exposed to Far-UV that would be dangerous? And have you thought about that?
Well, yeah, if you're not protected by the stratum corneum, then indeed cells could, living cells could be affected by the Far-UVC. So the issue of using Far-UVC, like in an operating theater, are a bit different from the issues of using it in a public location where you wouldn't expect to have open wounds.
And Stefan Jokush.
I have a question about oxygen, ozone generation. So basically, oxygen should absorb a little bit, and then it generates the O3.
Yeah, indeed, it does generate a very small amount of ozone, but way below the EPA safe levels. So it's a small issue, but I don't think it's a real issue, and particularly if it's being used in large spaces, not just in a very enclosed small space. We're going to be nowhere near the EPA limits on ozone generation or ozone